I'm Martin Brill, application specialist here from Rapp Optoelectronic, and we would like to thank that we get the opportunity to give a talk here at the Euro-BioImaging and at this TechExchange. And for the next 20 minutes, I would like to show you a bit what we can offer for advanced photomanipulation in biological research. So you might wonder, where does the name Rapp comes from? And it's from our founder, Gerd Rapp. He used to be a physicist, and he did his master and PhD in a physiology lab back in the 80s. And there he intended to provide a new device for photolysis to actually get a neurotransmitter at a certain space uncaged. So what it means, uncaging, here you see like a small sketch. You have a chemical that's in a chemical together. You use some strong UV light. It cleaves, and then you have a neurotransmitter like calcium, glutamate, or IP3. How does it look like? Here's like smooth muscle cells. People were interested. McCarran lab was interested in how these cells communicate via gap junctions. IP3, so inositol 3-phosphate, opens calcium stores. With calcium sensitive dye, you can now follow the calcium wave. So this is an application you can do with that. And just for completeness, Gerd Rapp is now at the retirement age. And a few years back, Sven Warnk joined the company, and he's now continuing the company. All right, back from the flash lamps, we also then developed devices for, since lasers became more affordable, more user-friendly, we use scanner systems, galvanometric scanner systems, and we have them in 3 flavors. That is a point spot, we call it Firefly, up to diffraction limited spot sizes. We can have like a certain module in between the laser and the scanner to provide shapes and spots of the laser beam to provide the stimulation. And finally, we have one of the best ablation systems in the market, would say, of course. And how does it work like? We add these devices to the back port, to the fluorescence port of microscopes, upright, also inverted. And yeah, that works quite nicely. All right, what can you do with that if you now use the scanners and we compare the same method or experiment we have seen before? This is now a single fiber smooth muscle, and you see at the tip of the arrows, there would be the uncaging laser. It's a UV laser, and you see this calcium influx IP3 opens the calcium stores. So this is one application we can provide. There's also deep UV applications that is able to be done with our system that is in cancer research, where you provide UVA and UVC DNA damage to the cell core. And what you see here is the fluorescence labeling. You see these 3 stripes, and these are PARP proteins, so repair proteins for the histone that got damaged. And they accumulate at these damaged places. So you can make a check what's better, what's less well done. Of course, typical photomanipulation would also be photoconversion here, like MEOS, where you flash the blue light onto the spindle apparatus, for example, and at the one-time hit, it changed from green into orange, and you can answer questions like, where did the origin of the spindle apparatus comes from? It's green, so something new happened there. Then, as I mentioned, ablation, we can cut actin filaments. We can see here typical applications like cortical laser ablations as validation for cell surface tension and interference of that, and even coarse applications where here development of gastrulation was analyzed. And one also very strong method that is used for protein dynamics is bleaching, FRAP, so fluorescence recovery after photobleaching. This is in a very small scale. This is an axon, and the people, Brophy lab from UK, they were interested in whether neurofascin is going retrograde or anterograde to the synapses. So you bleach once with a strong laser, you see the fluorescence is gone, but it comes back. And now with flip, so the fluorescence loss in photobleaching, meaning you continue the bleach. Now you could sequentially or both together see that there is no transport coming through. So that means this is an actual transport of this neurofascin. All right, these are some typical experiments or applications we can offer. In general, we provide applications from the deep UV up to the IR. And I already talked about some experiments or applications like DNA damage, uncaging, bleaching, but we also provide 2-photon ablation and T-jump experiments, heat jump, and so on. And there are more lasers to get. We have many of them. And depending on the application, we can provide systems for that. How do we do it? If we look into instrumentation, we are compatible with the main big 4 microscopic companies, Zeiss, Evident, Olympus, Leica, and Nikon, but also Sutter microscopes, Lux and Neumann, and Scientifica for electrophysiology, for example. As I mentioned, we add to the epifluorescence port, and what we are quite proud of, we have options to do this simultaneously with many of different imaging solutions in terms of like together with light sheet, super resolution, TIRF, and so on. And to show you that we can do this, here are some examples of advanced microscopy solutions. For example, we added our systems to Zeiss lattice light sheet, to the Zeiss ELIRA system for structured illumination microscopy, and there's nice work done now in UK from Vici Draviam from the Queen Mary University in London. We work quite nicely together with Evident. Here's like an example where we add our photomanipulation to the multi-photon system Fluvio 4000, but also with Nikon, with an Endo Dragonfly like here, or Leica STAT, or the Stellaris, and so on. And we are not even afraid if we get the request to put our system on even something more extreme. And that is, you might know it, the lattice light sheet from Nobel laureate Eric Batzik in Janelia, the HHMI Research Institute close to Washington, DC. And yes, indeed, if you take a look at the back of this system, there you find our photomanipulation scanner. So it gets in from the back and then is guided up here. What you can do with that, lattice light sheet makes beautiful 3-dimensional images. Here's a model, and people try to find out where the membrane and proteins and so on move to, and if there's some growth. And you see, especially with the FRAP, so that you do like a square FRAP here, you see that this structure moves up to the top of the cell away from the surface at head. All right, and enough from instrumentation. I also wanted to bring you some quite nice experiments, examples, let's say, for advanced photomanipulation. And there's one beautiful study from Kirsty Van from the University of Exeter in UK. And they did something fun. They work on annelid worms, the Platinerase. So it looks like this, but it's larvae in the 2-day stadium. It's like a roundish bubble, and it has a corona with cilia. And they were interested in actually how this pattern of movement coordinated. And what they did is they did ablations just of single cilia or a group of cilia and so on. And they could show that indeed, yes, this patterning is then disturbed. Quite fun to see. I love it every time. And another one from the EMBL, from Zinam Zaka's lab, spearheaded by Jennifer Heck and Christina Yevdokimenko, is DNA photolithography. So they used our scanner system to then draw a pattern onto conjugated DNA and then bind it to other DNA structures labeled with fluorescence is super beautiful, and it's a joy to watch these patterns. If we walk now from the UV to more IR side of the spectra, then we have here an example, FLIRT. So this is fast local IR thermogenetics. It's a bit like optogenetics, but instead of light, we are using heat. We can do this very special and confined with our scanners and with this device, the Geo module, for example. And what we see here, it's a thermosensitive mutant, myosin 2. And if you see there is no light in this control, then these cells undergo cell division. Instead, if you start the IR laser during this time, there's no cell division happening. It's quite significant. Or, of course, similar, we can have IR laser evoked gene operators that then switch on gene expression. All right. In the IR field, we also have a new device I would like to give some insight into, which works also with IR lasers. We call it flux focused light induced cytoplasmic streaming. It makes these beautiful images if you do a long time recording. And this was done in an endeavor together with Moritz Kreising and his group as a co-development, as he was at the Max Planck Institute for Cell Biology and Genetics, also together there with the light microscopy facility. This is a tool to manipulate samples. It's non-invasive and different, but similar to optical tweezers. And it works by moderately heating the specimen. How does it look like? What can we actually do? We can make use of our knowledge of photomanipulation software and knowledge of how we control lasers. And what you see here are actually fluorescent beads in a honey solution. So that resembles the viscosity of cells. And you see that we can control the direction and the speed and so on of the particles in this solution. And this works quite nicely. It works through thermal conduction into different streams that elicit from this different heating. We can maybe discuss it later. Right, and now the question is, of course, that's fun to see, but does it have any biological implication? And for that, yes, Moritz Kreising's lab, what they did is they work into development. They can rotate the PAR domain of this C. elegans embryo. You could even roll and turn the cell core of embryos, and it's strong enough under certain conditions that you can even shear off or cut off the spindle apparatus here in this embryo. An amazing technique, and we are happy to provide you with some insights or devices. All right. Beside this, we are also working in the IR regime, but with ultra-fast femtosecond lasers, mainly 2-photon imaging. And I guess you have already heard what's about. You only have the fluorescence elicited in the focus, nothing above or below, just IR. It's perfectly suited for in vivo work. It is ideal for deep tissue with high resolution imaging in vitro and living animals. We use different types of ultra-fast femtosecond lasers. We have a nice developing group here to make this. You use a 2-photon microscope and get these nice images here from Ana Sanchez, for example, from Plested Lab in Berlin. Our customers then obviously do in vivo work. That could be the fruit fly. We have a customer just setting it up for ants now, or the axolotl, and of course, nice mouse work that has been done. What does this have to do with photomanipulation? Of course, we can add our photomanipulation devices. I already showed you our beam pass. Now it's getting a bit more complicated. So we are actually also resellers for Shutter microscopes, and we provide customized 2-photon microscopes. So from the shelf to the table, microscope, computer, everything. And here, this is an in vitro microscope for slicers or organoids, so that a 1-photon photostimulation is okay. We can scan it here with up to 6 different laser wavelengths. It goes through the same objective, and this system is then meant for recording and acquisition of the reflected 2-photon elicited fluorescence and for the transmitted 2-photon elicited fluorescence. Apart from 1-photon photostimulation, we are also proud to share that we can provide holographic stimulation. That is a stimulation, a multi-site stimulation, if you will. We use SLM-based systems. Here with the other nice microscopes you can use for in vivo work. It's the MOM from Shutter, and we can add the holographic stimulation. You see this box on the drawing here in the back. This is one of our calibration views, which we use for automatized calibration. We again make use of our knowledge of photostimulation software. As you see, different patterns here and different time strains. We can have different sizes for the stimulation. And how does it look in a fish? In this example, we got these nice samples from Ari Arenberg's lab to test it. So this is convertible GFP in DPF fixed zebrafish. And we can provide now these patterns in different sizes in X and Y, in different Z sets. And you can use it mainly for optogenetics. Depending on the laser, it could also be used for stronger applications, but it's meant for optogenetics. And with that, I could show you that we offer a vast variety of applications for biological research that spans from deep UV up to IR. We are not afraid to do customized solutions. Right now, UV microscopy, wide field or stimulation is quite on request, and we can offer solutions for this. You see here our ablation system. This is actually my hair, which we engraved with the name of the product line, UGA42. And yeah, for that, I'm happy to answer questions if there are some.